So I'm interested in phenotype-genotype connections for my multi-drug resistant clinical E. coli. So why do we care about antimicrobial resistance? So throw some statistics at you that you've probably already seen 1,000 times. 2019, AMR was directly responsible for nearly 5 million deaths worldwide, and an additional 1.27 million were attributed to AMR, not directly caused by it, but and then as of 2050, I'm sure you've all heard, it will be causing 10 million deaths per year. And that has a massive financial burden on healthcare systems, as seen by that figure at the bottom. 2.7 billion is a lot. So yeah, what do I mean by phenotype-genotype connections? So resistance is an evolutionary process, as it says, and fitness in both the presence and absence of antibiotics affects that prevalence. So if you've got a mutation or a gene that is more costly, the prevalence of that organism with that mutation or gene is going to be a lot lower than something that does not cost as much. And predicting the spread of multi-drug resistant strains requires an understanding of that connection between the phenotype that we observe mainly in the lab and what exists in its genome. So my collection is a collection that we call the Wythenshawe strains, and it is made up of 91 clinical isolates of E. coli, all collected from Wythenshawe Hospital, which is somewhere around here. And we don't know what the sample sites are. So I get asked a lot, where have they come from? I don't know. UTIs, bloodstream infections, could be anything. So these are multi-drug resistant. I know that because I've tested them phenotypically on clinically relevant antibiotics. And we also sequenced the genomes, which Microbes NG did fantastically. So we're looking at genotype-phenotype connections in clinically resistant E. coli. We looked at, like I said, resistance phenotyping. We did this qualitative. So yeah, on agar, stamp it on. Does it grow? Does it not grow? Using these breakpoint values that UCAS, lovely company, publish every year. And we did it quantitative, so looking at fitness, growth over 24 hours. We did a bit of genomic AMR profiling. I'll come to the end of the pipeline that I used for stuff like this in a second. We did phylogeny, we looked at gene presence, absence, and finally the association between those genes and those traits. So this is my pipeline. Although I realised that I must have forgotten something off this pipeline. So basically, I take those lovely sequences that Microbes NG have given us. I reannotated them because they were missing some of the genes that I needed. So I reannotated them using our MG1655 E. coli reference genome. I predicted the pangenome, so gene presence absence, and I associated those genes in that pangenome to the phenotypes that I saw. I also did a bit of resistance gene profiling and looked at SNPs and used that core phylogeny to make, a tree, which will show up at some point on here. So my first question was, is there a cost of multi-drug resistance? So the more resistant genes, mutations you have, is it more costly in the absence of antibiotics? The answer to that question is no. There is no association between fitness in our multi-drug resistant strains and the amount of resistance they show phenotypically, which is quite worrying in the grand scheme of things. And so then my brain started to go to, well, some organisms have more than one gene that confers resistance to a single class of antibiotics. Is there a better, fitter gene that exists? So for this, I narrowed my antibiotics down to 4. So I looked at cefpodoxime, which, if you don't know, is a beta-lactam. So we were looking for beta-lactamases there. Aminoglycosides, so we used gentamicin for that. Trimethoprim, because who doesn't love trimethoprim? And a SNP-mediated resistance, ciprofloxacin. So we found that there was no consistent advantage of having certain resistance genes. I expected them to cluster and look like there were some that performed better than others. and that just didn't happen. And even those that have several resistance genes, as you can see by the cool little pie charts at the top where they're split, performed better, but it wasn't really consistent, and there's not really enough there for me to make a conclusion about multiple genes that exist within a genome. But what we did see that was cool with ciprofloxacin is that these ones that have a triple mutant, so the green hurt your eye colour, have a gyrase mutation and a PARC, two gyrase mutations and a PARC mutation. That triple mutant makes them really, really good at growing in the presence of ciprofloxacin. But it doesn't deter how well they can grow in the absence of antibiotics. So higher fitness in the presence of the drug, no obvious fitness cost to that mutation, which is funky. So then I thought, well, we have all of these genomes, let's look at how diverse these genomes are. And so we did a bit of ST typing. So everyone will know, but the most common one that we think of when we think of E. coli infections is ST131, which is the nice clade in the middle, which surprisingly all of those CYP-resistant ones belong to. But yeah, you can see that the Wythenshawe strain collection, as is a very diverse collection. There's a lot going on with it, and that is just confirmed even further by the pangenome of it. So the same tree, gene presence, absence, so everything that is going on in these E. coli genomes, and just so diverse, even in the same clade, so diverse. So then we can start looking, well, what are we trying to ask by looking at, everything that exists in a microbial genome, along with these phenotypes? What genes, if not resistance genes, are associated with resistant phenotypes? How much of the functions of those genes are distributed within the genome? Are those gene functions associated with the mechanism of action of that antibiotic? So again, for this sort of section, I'm going to be focusing on those 4 antibiotics just to keep things consistent, because if I did everything, we'd be here all day. So why do I care about gene function? So I want to understand the gene function and the interaction between those genes and antibiotic resistance genes. Yes, it's not in alphabetical order. Ask NCBI why it's not in alphabetical order. But the ones that we're interested in are these, what they're classed as defense mechanisms. And Will tell you they're not defense mechanisms, they are antibiotic resistance genes. But that's what they're classed under using this system. And the system that I used was COGS, which are clusters of orthologous genes. And that assigns basically a little ID to each of your genes, and that will give it a functional category, so kind of the function of it, and then you can kind of group things by what their function is based on that. And they originally started off as an evolutionary classification of protein families, but they've widely been used now in this sort of functional genomics story. And I forgot this on the pipeline, which is where that came from. I used this cool program called DeepNog, which is a version of a program called Eggnog, which I'm not sure if you've heard about. And that just, you feed a load of proteins into it and it'll assign those IDs for you. And so I'm just going to fly through this because, to be quite honest, I've made this less than 24 hours ago. And so who knows what's going to go on. We're all on this journey together. So this is the entire pangenome of my E. coli and all of the COG categories that exist in them. I'm going to highlight some important ones because I'm not going to go through all of them because, again, we'd be here all day. The V at the bottom is the defense mechanism, so the resistance genes that exist in it. We've got the mobilome, which considers phages, transposons, integrases, all of that sort of jazz. And the highest one in there, which I found quite surprising, was carb transport and metabolism. So quite diverse across the pangenome of things that exist within those functional categories. And so my question is within those antibiotics, what is the distribution of these functional categories for genes relevant to those phenotypes? So we can look at cefpodoxime. We know the action of cefpodoxime. It affects cell wall biosynthesis. The resistance mechanism is through beta-lactamases, extended spectrum beta-lactamases, carbapentamases, and we can see here that quite a large quantity of the mobilome, which I would kind of consider to be relevant because those genes are most likely plasmid associated. So yeah, makes sense. Again, you've got amino acid transport, cell wall biogenesis, all of those things kind of link into it. We're going to fly through it because I've got 2 minutes left. We look at gentamicin, negatively impacts protein synthesis. It does it through ribosomal inhibition. Don't exactly know how. Efflux pumps, again, signal transduction, cell wall biogenesis, transcription, all affected in sort of associated with that phenotype. Trimethoprim, which is my favourite, is involved in the folate pathway. It interferes with DNA synthesis, so it is mutagenic. Dihydrofolate reductases, efflux pumps, again, kind of enzymatic. And our key ones here, which is fantastic, is transcription, replication, and repair, which is amazing. Again, I've highlighted on all of them, but I've just flicked through really fast where the resistance genes lie. So in kind of a distribution. And actually for trimethoprim, the resistance genes quite high frequency. And then we look at ciprofloxacin. This one's funky. What is it doing? So you'd expect, with ciprofloxacin being SNP mediated, you'd expect kind of the genes associated with that resistance mechanism to be transcription, repair, you know, a bit like trimethoprim was. But no, what's the mobilome doing? So that was fascinating, and I found that out this morning. So I would love to know what's going on there. And then again, we've got this R, this peak in R, and R is a general function. They've not had a specified function put to them yet, so I can't really make a conclusion about what's going on with this one, but I can kind of look around it. So my conclusions for this is resistance genes are responsible for resistance phenotypes, obviously. There is a V on every single one of those histograms that you saw. Yeah, so we know that resistance genes are associated, they are relevant to that phenotype. But in most cases, there are other genes associated with those phenotypes, and how much should these be considered? And how much do they affect, like, say things like MIC, things that we measure in the lab. And on a more personal note, what's going on with ciprofloxacin resistance? Who knows? And so I'd like to thank everyone that was involved. And we do have a new Merman logo that Zara made somewhere in the back. But I forgot to add it in. But it's on your badge, so look at it. Yeah, so thank you. Thank you for listening to me talk.